previous two sessions that we had seen is that mostly we were looking at the genomics based studies, how the so why the how the omics the rapid sequencing of the genome and the whole genome based studies have led to the identification of the different uh, copy number alterations, variations, and all. But we don't get the whole picture of how it is being actually reflected at the person. For example, a small example of what we know is that the mostly the genes and the genomic structures remains the same for different organisms, but each and every organism behave differently. So though we have studied the different genes and we have understood the different mutations and the different genome level alterations, but we need to go for at each and every step of the central dogma to have a specialized study to find out the different levels and to get the whole picture of the cellular level processes that are happening. So moving on, so each and every step of like the DNA, so the omics or the nomenclature of the omics, or if we say that the like we are studying the proteogenomics, but the multi-omics, or so what is the so nomenclature stands like this, like that the for the DNA-based study is the genomics. At the RNA level, if we study that, we have seen that the abundance level is the transcriptomics. For the protein, if we try to find out, and the omics means that to study the whole the each and every gene, each and every protein, or each and every transcript level in a particular cell or a particular body. So this is the proteomics, and further is the metabolites. So metabolomics. So all the chains that goes on in a structure that frames any person, any living organism, and the study of those is known as the proteomics, where we are focusing mostly on the proteomics and the genomics technologies. So now, as I was mentioning, that the DNA of the human and the mouse and the chimpanzee. So here you can see that the human and chimpanzee share a surprising that it is almost 98.8% of their DNA is same. But as I mentioned, that only genomics knowing, as people were thinking, as scientists were thinking that after going for the human genome map and identification of all the genes, that it is enough for us to understand that what alterations and what changes are being going on, uh, what or things are going on, but it is not the case. So why is that? If the number of genes are quite similar for most of the organisms, starting from a small model organisms, you can think of like C. elegans or mouse or butterfly, that the number of genes, or the human like complex organism like human, that the are mostly are quite similar, but how still all the organisms are different, as well as within our organism particular. For example, if you take for a human, that if you take heart, if you take lung, if you take other organs, they are quite different in function and the structure. So if the number of genes and the DNA content is same, then how this happening? So that's why understanding different omics technologies are required, and we know that there are various factors from DNA to protein. While making in the from the molecular biology, we know that we need alternative splicing, post-translation modifications, etc., which gives the variety of the mRNA-based transcripts, and which further goes into the proteins. So that's what make us quite different, and that's why it is required. So this is a typical pyramid of a integrated view of the cellular processes. If we look at it, so here what we is that, like the genome, it's quite static. So genome, we can see that it is quite static, and it is in the top level. And also if we go on in the complexity, so complexity goes on increasing as we go below with further dynamics and all those things getting added, the various modifications and all getting added as we move down. So genome is quite static. Then there comes with some epigenetic variations, as we have looked at the previous class, the different epigenetic variations, that what happens and let us the DNA, like the methylation and the modifications and the acetylations, led to different kinds of epigenetic interactions. Then further moving on to the transcriptome is there is the alternative splicing, as I mentioned, and which leads to the variety of the protein structures. And finally is the proteins, which are due to like the phosphorylation, acetylation, and there are uh, varieties of PTMs and all, which leads to the different functions of a protein. And also it is important for the to maintain the structure and the integrity of the protein. So the modifications helps to protein to retain their normal structure and the function. Without those modifications, that the protein won't be able to function. So this is an integrated view of the cellular processes with the genome to the proteome, and going with the different classes of the complexities and getting much more complex and diverse. Now we will be looking at today is that after getting an initial idea of the genomics based approaches, how the genome is functioning, how we can sequence those genomes, and how we the different quality check is required. Then follow how the different synthesis sequencing technologies, like from the Sanger sequencing method to your NGS, and now the Oxford nanopore sequencing method. It has helped us to, in every step, it has helped us to sequence it in a rapid rate at a low cost, and helped us to get a maximum coverage and to gain insights regarding the various genome level things. Also, the human genome map was a very much we know that the human genome map was a very milestone in the field to help boost this kind of research and study, and now it is being extensively used for identification of the mutations in the cancer, in the disease patient, in different varieties of things. So similarly, the proteomics approach, that in that decade only, and after that, it was needed to understand that what is the level of the protein in the human genome. Though many people that time was, as it was mostly related to the genomics based studies, but there have been some uh, further advancement in the proteomics based study as well to identify the proteins in a particular human. So these are the two milestones of the papers of the journey, where you can see that mass spectrometry based draft map of the human proteomes, that what proteins are present in the human body, in the both using organs, all organ based proteome map was developed, and it was they used the mass spectrometry. So the advancement of mass spectrometry helped, like the NGS helped to have a very good rapid and the sequencing. So mass spectrometry helped in high throughput advancing and the sequencing of the identification of the proteins, because you know that. So if we go by the history of the protein sequencing technologies, if like the NGS or the genomics sequencing technologies, so proteomic sequencing is like the Edman degradation. So that was not at all very much high throughput, and the identification was quite not so much robust. So mass spectrometry has helped us to uh, take us to a new level to identification of those proteins. So the mass spectrometry was proteomics has helped helped us in great insight in the proteomics advancement of the proteomics field. So another, like the human protein atlas, a special map of the human protein. So this is another which has tried to map the human proteins using the antibody based approaches. So they are, as you all might be aware, that the ELISA, Western blot, and all are the different antibody-based approaches are generally used in different clinical or diagnostic settings, as well as for the uh, antibody for the validation of the particular protein. But there is a slight difference between the antibody and the mass spectrometry, with advantages of both, advantages and disadvantages. Like antibody based is quite you need to have a is quite extensive, intensive, labor intensive, as well as the cost. You need to have the antibody, you need to have the proteins. For that we need to raise the antibodies, and then only you can do that. Whereas mass spectrometry can help in identification without and in a high throughput manner without the use of those particular antibodies. So there are so I recommend you all to go and try to figure out that advantages and disadvantages of the both mass spectrometry based technique as well as the antibody based techniques. What are the advantages and what are the disadvantages for each. So this is one of the database which is Human Protein Atlas. So they have made the resource for the like the tissue type uh, proteins, tissue type proteins, brain enriched the genes or the proteins that are enriched in the particular region. So as of now, we used to know that, as I mentioned there, that the different organs in a human also, not only different organism but the same human beings, the same different organs function quite differently. So there are a set of proteins or the genes that are enriched in a particular region but not in other regions. So what are those? So you can go and try to find out in the Human Protein Atlas, which will give you to the different sections, single cell type, tissue type, disease type, and all the proteins how they are expressed. So so as I said, that what is the antibody based approach, and how the mass spectrometry based also uh, based approach, what is the mostly the different, as well as the mRNA level transcript level abundance also you can try to find out. Then also you will get a comparative understanding that how your from the genome the transcript level changes and finally the protein level changes are happening, and which is forming the current organism or the state or a normal physiological state, whatever understanding. So why I put out this thing is to draw out an analogy that your globe which was present, your globe is present from a variety of time, but can you, if just think or imagine that, can you try to use this for navigation? No, right? So you cannot take out a globe and try to navigate to the different parts of the world. But with the advent of the Google Maps, you can easily go and navigate in each and every street and corner. So the resources like Human Protein Atlas and all, with Uniprot, with all coming up, it has helped you to understand and it has helped you to uh, kind of navigate, kind of understand the specific expressions in a particular disease state or in a particular gene state or particular in a disease state or a particular cell type, that how you can see and visualize the things. So that's why this kind of resources are quite important for researchers, for scientists to understand that what is the exact level changes, and then further go on investigating the causes behind any abnormality or anything. So that's why I try to draw an analogy that why, how in these fields the advent of data to in an open access manner is quite important for anyone to go further on the different studies. So this is the how sorry. So this is how the different field as genomics technologies got evolved, proteomics technologies has also got evolved. So if we look at first instance of proteomics tree, that what kind of analysis, what kind of study, what kind of insights, biological insights we can get. So for example, the proteome mining or the global proteomics based study, we got the discovery based proteomics, if you want to say, that you can get the drug discovery, the targets for drug discovery, the targets as a biomarker, as a which in a two states, which markers are coming as a uh, differential expression, showing differential expression, showing truncated expression, and all due to the various reasons before the protein expression. So you can find out those. Another aspect is the functional proteomics. Then you can identify the structural, the protein complexes, then the protein-protein interactions, that with which protein and the structures and the interactions that play a role in certain expressions, then the protein expression profiling with the signal transaction and the disease mechanisms, and the PTMs as well. So proteomics, under the branch of this proteomics, you can try to understand all this, like the phosphorylation glycosylation based PTM identification using mass spec. You can do that. You can do that to find out the different. So mostly, generally, mass spectrometry based proteomics helps to understand the protein expression profiling and the proteome mining and the PTMs and the protein and the PTMs post translational modifications. Whereas the protein interactions like the SPR and all those helps to understand the in the different interactomics, I mean, interactions based studies one can understand. But the mass spectrometry based proteomics mostly helps us to understand the differential expression, the PTMs, and the in the different states. So this is how we can study under the field of proteomics. As in the genomics, we have studied the whole genome sequencing, whole exome sequencing, copy number alterations, the variants, effect of those variants. Similarly, in the epigenetics, we had looked at the different genome-wide association studies and all those interactions. Similarly, under the branch of proteomics, we can study all this field. So, is there any question? No, right? So, so we can move on. So, in this proteomics, mostly what is before the mass spectrometry, and also there are actually, we can say that in a rough manner, to divide it into two way. One is your gel based proteomics and another is the gel free proteomics. So gel free proteomics, by the name you can understand that do not take in the use of gel or SDS based gel that is generally being used for differentiating the proteins on the basis of their molecular weight. And so yeah, and another was the gel based proteomics. So before so without the use of mass spectrometry or any of these high throughput techniques, so the 2D uh, dye has been used into many extent to find out the differential expression of the particular proteins. So similarly, so we know that the, so in the basics that the SDS page gel uses the separation on the basis of the molecular weight. So I hope that you all are clear about the principle, where we take the, on the basis of the acrylamide, the stacking in the dissolving gel, the proteins gets resolved on the basis of their molecular weight, and we can visualize those by using different stains. So different stains gives us the proteins that at what molecular weight. But there might be cases that the proteins have, different proteins have the same molecular weight, and that do not give a better resolution or cannot be resolved it properly. So then came the concept of 2D gel electrophoresis by O. Farrell, which said that first dimension separation on the basis of the pi. So you know that at the isoelectric point there will be no net charge on the protein and there will be no net movement, and followed by first dimension separation. Second is the keeping that uh, IE strips onto the gel and separating it on the basis of the molecular weight. So once one dimension it is being separated on the basis of pi isoelectric point, second dimension it is being uh, separated on the basis of the molecular weight. So this has provided and helped to give a better resolution in the gel and better uh, into identification of much number of proteins on the basis of both the things. And third is that the 2D diet differential, which takes in account the principle of 2D, but helps into understand and identify the differential expression of the proteins. So how this 2D diet work? So if we looked at, so now if you have a protein extract from condition 1, so you are and 3 replicates. For each condition, you have 3 replicates. So let us now try to understand that how the with the help of 2D dyes you can try to identify that what is the differential expression of the protein. So just try to understand that first you have the two conditions, and in each condition you have three replicates. So for condition 1 you have three replicates, and condition 2 you have three replicates. Now you have extracted that protein through different means, and for that condition, for
So
condition 1,
I didn't understand the DIG thing.
sorry,
What replicates are you talking about?
what can you repeat it once again, please? Become very
सर
slow your
मुझे वो
voice.
replicates का नहीं समझा.
Okay, so here you have condition 1 and condition 2. Okay, and in condition 1 you have three replicates. Okay, and condition 2 three replicates. So generally when you do any experiment, so generally what we do is that we take control 3 and treated 3, n equal to 3 for control or n equal to 3 for treated, or 3 patients control, 3 healthy controls, 3 disease patient. Okay,
Are you talking that we are doing the experiment in triplicate?
yeah, for example you just consider that. Okay,
Yes.
so now for the, or this is actually the biological replicates, but here actually what I meant to say was if here what you will do is that you will so these are there are two replicates actually. So one is your biological replicates as I mentioned, and one is the technical replicates where for each person only you will run 2 or 3 times. Okay, so don't just go into those complexities. Now let us just look at it how it works. So you have protein extract of the condition 1. Okay, you just take at don't go over the biological or the tech, just think that there are three tubes with the condition 1. You have labeled with psi 3, and you have three groups, three tubes with the, you have labeled with the psi 5. Okay, so psi 3 and psi 5 are the two dyes which will fluorescence and will give you the result. Now what you will do is that with those three tubes and with this three condition 1 and the three condition 2, what you will do is that you will label it. Okay, so now what you will have done is that you have pulled internal standard with the psi 2, correct? Now you will do the, you will run this in the gel, both psi 3 psi 5 and the psi 2. Psi 3 is for condition 1, psi 5 is condition 2, and psi 2 is the labeled with the internal standard pool. Now after running you will scan in three different wavelengths to get those results. Now why this so now you can see that you will get like the protein expression after you perform the analysis and all. Certain protein level changes in certain parameters, and certain protein level changes in another condition. So now what is the role of this psi 2? So since you are doing the gels and the experiments, so what happens is that there are some technical variances that occur that will lead you to does not understand that how much you need to normalize, or to, if for a differential expression you need to have the, if you want to compare the differential expression between the condition 1 and the condition 2 and say that this protein is upregulated or this protein expression is downregulated, so you have a reference that both the expression is same. But what happens in the gel based experiments is that there are sometimes certain spots you will find out that certain it has got some down or some up. So from there to take out or quantify is quite difficult. That's why psi 2 is the just now focus on it to understand that the psi 2 contains all the pooled internal standard. So the psi 2 is the same sample. Okay, technically psi 2 is the same sample which has been run in all the gels. So the psi 2 labeled proteins should come in the same spot in each and every gel. So if you run 3 gels, if you run 6 gels, the psi 2 labeled thing should come in the same in each and every gel. So on the basis of that psi 2, the it is normalized. Okay, so for psi 2, for example, protein X has moved, not protein X, spot X has moved to certain position by x x millimeter or something like that in the software, that the psi2 spot has moved that. So for both psi3 and both psi5, that will be moved exactly the same way. Then you will get that, okay, so all the technical variances have been reduced, and then you can follow the you can try to find out the differential expression. Okay, am I clear to everyone?
No, sir, I don't get it.
Which part you didn't get it?
Psi 2.
So psi2 is, so as I said, that you have 3 tubes that you have labeled with psi3, and 3 tubes you have labeled with psi5. Okay, so now you have 6 samples. Okay, and now you take all the aliquots from each sample and you have labeled with psi2. Till now clear?
Yes, sir.
Now it's clear, right? Now you are running 6 gels, right? For 3 conditions, 3 tube, 3 gels,
Yes,
3 conditions, 3 tubes, 3 gels, right?
yes, sir.
Yeah, so now is that if we now each and every gel, there will be slight differences. If we run 3 gels, it will get that there are slight differences, but we cannot compare it with. But psi2, since it will be run in all the gels, so we will get a reference point that how much technical variances is there. So Sci2's work is to just to give us the reference that how much the technical variances are there, so that we can just we can just move our spots accordingly for Psi3 and Psi5, and just can take out the differential expression. I hope I'm clear, right? Okay. Yeah. So this is for the
Yes,
Dij.
sir.
Yeah, identification of the differential expression of the proteins. So it's nothing, uh, just psi2 is the reference, which is psi2 means the proteins. First you will make the pool of those particular proteins, and then what you will do is that you will die with the psi2, and then that generally with the psi2, and then you will take the reference to get that how much there are technical variances, and then move accordingly the spots for psi3 and psi5, and just identify the differential expression of the proteins. So this is the concept of the internal standard. So this internal standard, the pool internal standards, are actually helpful for any proteomics based experiments. For example, in this DIGE, it is quite helpful for us to understand, to give the reference, since it is technically the same. Similarly, in the mass spectrometry based approaches also, it is quite recommended and to have a pool samples, which gives us the variety or the technical variances that are happening, so that we can get an idea, because these are the same samples. So one sample, if it is run 100 times, should give the same thing, but it doesn't happen in reality because of certain technical various things are here. So that is the concept of the internal standards. So, but what was the problem of this 2D DIGE is that it is low throughput and has some issues with the reproducibility, since you know that running the gel and all, first labeling and then running and then scanning, all these things is quite problematic and have a lot of steps which can be with the formation of the gel can be faulty. There can be voltage issues while running your samples, and there are multiple things that can be cause trouble. So these are there are certain issues with the reproducibility and its low throughput. But one application of 2D dye which is still in use can be said that the host expression system, identification of the host expression systems, when using in where, for example, if you are doing those yeast and all cloning, and also what is the percentage in the host expression systems and all, there are certain things where 2D dye is being still used to identify those. So now moving on, so after the gel based proteomics, so there was the gel free proteomics, which is mostly used in the mass spectrometry based proteomics. So so I hope it is clear till the gel based 2D and dye, and I think there's no issue, right? So now we will move on to the mass spectrometry based proteomics. So what happens? So here it is the one kind of basic diagram, which is nothing but firstly, if we inject the samples, then how it happens is that it gives the ions are formed in the gaseous phase, and after those ions are formed, it is accelerated on the and separated, and then finally, so different mass analyzers are there which separates those ions that have been formed, and after forming the ions, it is detected, which gives us the signals that what ions are generated. And so generally what we measure in the mass spectrometry is the mass per charge ratio. So finally we can identify that what is the molecular weight of that particular sample that you have injected, and then you can trace back. So we'll be going into the detail in the subsequent slides. So this is just an broad overview of how mass spectrometry concept, that going on to the generation of the creation of those ions, acceleration and detection using the ions, which has greatly revolutionized this field of proteomics and has led us to form so much high throughput techniques. So this is how generally what we perform the mass spectrometry. So for example, you get the tissue from any or a tissue or any cell lysate, or you bring any plant cells. So what you will do is that you will extract out the proteins, and then you will digest into the peptides. So this is an approach of our this is known as a bottom up proteomics approach. So we will look into it detail. So just from now on, just uh, be careful in certain terminologies and all those, and if you cannot understand anything, you can say. So these are the peptides. So mostly our peptides are in hydrophobic in nature, and different proteolytic enzymes are used. So different proteolytic enzymes like trypsin, chymotrypsin, lyse, these are the different peptides. These are different enzymes that are being used in digestion or digesting the peptides. Then a liquid chromatography is being placed because you know that so many peptides, if you give it at once to make the ions and all, it will be, the detector will get bombarded, and it will not able to, there will be overlap, and you cannot identify those peptides or in a very well fashioned or well mannered. So you generally what we do is that a reverse phase liquid chromatography is being followed, where I think that what is the principle of the reverse phase. So if you know that in the reverse phase there is a column where where the hydrophobic peptides gets bind to it, and after that in the by the mobile phase with certain increasing the hydrophobicity, the peptides gets eluted out, and then finally taken into the mass spectrometer. So in the mass spectrometry, so in the reverse phase, what so in the after this liquid chromatography, when you have eluted out the hydrophobic peptides uh, with the mobile phase, then what you do is that when the ions are generated, then the mass analyzers separate those ions and fragments those, and then you get the MSMS spectra. So this is a general mass spectrometry based proteomics workflow. So if you are not clear with anything, so don't worry. So we'll look into it in more detail in a stepwise manner. So this is a general workflow what is done. So the protein is digested into peptides, separated by liquid chromatography, then ions are created and then are detected in the mass spectrometer. So how this is done? This is so you all know the peptide structure, that peptides are formed by the CONH bonds, and there is the side chain, and then it is finally the peptides are linked together by the CONH bonds. So there actually what happens is that if you go by the peptide structure, so there are A, B, C, and X, Y, Z ions. When the ions gets fragmented, So these ions are generated, but we use a proteolytic enzyme and which helps you to gives us which cleaves at the C terminus. So this leads to the mode fragmentation of the Y-type ions. So generally in the mass spectrometry, when we try to identify, so mostly the ions which are cutting or fragmenting at the uh, C terminus is the Y ions, and in the N terminus is the B ions, which is generally known by the terminology, which because the other ions, other cleavage sites are called the XYZ, and the other cleavage sites are called the cleavage, not the cleavage, the fragmentation sites are called the ABC ions. But mostly in the mass spectrometry, what we get is that the Y ions and the B ions are the most prevalent, uh, and the C terminus cleavage and the N terminus cleavage. Among these also, we get the Y ions cleavage more because of the use of different proteolytic enzymes. So how it happens is that, so the basically Y1 should be Y1 and B7 is a pair with the cutting of the same amino acids. Similarly, Y2 and B6 will be a pair. So now if you want to identify that what is that particular amino acid in between those two, so what you will generally you will do is that you will minus y1 minus y2 and b7 minus b6. So if the all the ions and the fragmentation have been done at 100% efficiency and the ions have been generated and detected, then y1 minus y2 should be equal to b7 minus b6. But in reality, what happens is that mostly the y1 and the Y ions are only prevalent because of this kind of the use of the enzymes, and B ions are not so prevalent. Okay, so this is the how what if you just need to understand that the peptides, what ions are generally generated in the and detected are the Y ions that are getting detected. Is there any question or anything? No, right?
So how come Y1, Y2 is equal to B7, B6?
Sorry,
Sir,
can you repeat it?
how come
Hmm.
Y1, Y2 is equals to B7, B6? I didn't
Now
understand.
y1 minus y2 equal to b7 minus b6 because this is coming from the this is the same pair. Y1 and b7 is the same pair. Y1 is of the C terminal fragmentation and b7 is the N terminal fragmentation of the same site, same amino acid residue, and similarly is that y2 and b6. So by minus y2 minus y1 and minus b6 minus b7 shall give us the same mass of the same particular same amino acid. So we will go on mostly to the mass and mass plus charge, what is that and how we will, then I think it will become much more clear. So now for now you know that the C terminus fragmentation is the Y ions and the N terminus is the B ions. Okay, so now if we go for the quantitative proteomics, so now as I mentioned that in the gel, in the 2D dyes, you can get to understand that what is the two differential protein expression in the healthy and the disease state, or in different conditions, whatever, if you treat your cells in certain conditions and if you treat the cells with other conditions, for example as you design your experiment. So similarly in the mass spectrometry based approaches as well, we can understand the quantitative proteomics. So we can quantify that how much is in the expression level. So that's what comes into, there are two kinds of quantitative proteomics. One is your level free quantitation, another is the level based quantitation. So what happens in the level free quantitation is that there is no other kind of any kind of labels are being added in the peptides. Just the so again you can get a glimpse of the workflow of that, how the peptide, how what is the workflow of the mass spectrometry proteomics. So you can take the protein extracts, you have done the trypsin peptides, and then you can quantify without the on the basis of the spectral counts, and do not need to have an any labels or any uh, ions to identify that, for example, this intensity and this intensity is quite different in the control versus this. So this is and identify the LCM SMS. In label-based quantification, is what happens is that you labeled your peptides with certain uh, ions or certain labels that doesn't change the mass for any of those particular disease, and on the basis of those reporter ions, on the basis of those ions intensity, you try to understand that in each condition what is the difference between those particular proteins. So this is just you can understand that these are the two methods for quantitation that are being in use. So, for example, here what happens is that in one of kind of the label-based quantitation approach is the ITRAC or isoperiodic tag for relative absolute quantification. It's full name. What it is known as that you have the sample rate up to sample rate. So what happens? What major difference between your label-free approach and the label-based approach is that label-based approaches are quite, you can multiplex your experiment. That is that in a single run you can try to identify your different 8 samples you can run in. You can have a single mass spec run and can try to identify the differences in the 8 samples at once, whereas in the label free you have to run each sample at once, and then only you can try to find out that what is the difference between your healthy group or the disease group or any two plant species groups, etc., anything. So you need to have each run, but where in the label based you can quite do a multiplex. So just try to understand and these terms, like the multiplexing helps can be done using the label based approach. So here what you will do is that you will add the reporter ions and the balancer ions. So this balance group is 28 to 31 Dalton. This is done on the basis of, since you can see that the reporter ions level are 114, 115, 116, and 117. So on the basis of that, there is a difference in the mass Dalton and the balance group as well. So then overall mass of the reporter plus balance comes as constant. So these reporter ions or these label ions doesn't add any extra mass to your particular sample or particular protein, but their intensity will, you can try to find out their intensities, and then you can understand, since you are labeling your sample 1 with 114, so you know that my this sample is been labeled with this one and this sample has been labeled with another reporter ion. So on the basis of those reporter ions you can try to find out that okay, so this reporter ion is has this much intensity. This means that this condition for particular this protein, this is upregulated or downregulated. So this, in this way you can try to find out what is the differential expression. So this is the label based approach. And then is this generally the label free quantification or the label based quantification, whatever is happening mostly on the basis of the discovery based work. What is your discovery based work? Means you have been given two conditions to try to identify what is the difference in their proteome level expression, and then you are trying to find out that okay, so this has the proteins that you have identified on the basis of your experiment, that these are that are differentially regulated. Now if you try to find out that okay, these particular proteins is only is the correct one, and if you want to validate it, so what you do is that generally you give the mass spec that okay, I want to search this particular precursor and this particular fragments of it, and say that because you know the proteins and the peptides, and you can generate the theoretical fragmentation pattern. So if you don't understand anything now, just wait, we'll go into it in more detail, but just try to get familiarized with the certain terms. So we know that the particular peptide which we are looking to and which we are trying to target, so they are fragments and they are uh, next precursors and their fragments, or these daughter ions. So we will tell the instrument to monitor those ions only, and if it is those ions are present in our sample, then it will get detected. So for this mass analyzer, like the quadruple mass analyzer and the triple quadruple mass spectrometer is being employed, which where it generally acts as a filter for those particular precursor and the product ions that you can further detect. So if we, so I don't know if you just to let it begin from each and everything. So the skeleton of the mass spectrometry is that what is first is the creation of ions. So just try to understand in a simple way is that you need to, so how it works is that firstly the ions are created, then the ions are separated, and then the ions are detected. So these are the three simple steps, as I mentioned in that also, in that that diagram, that how the mass spectrometry generally works. If these are the though I don't know if you think that, if you feel that it is quite complicated, but the three just simple steps is that creation of ions, separation of ions, and the detection of ions. So what happens is that firstly the ionization procedure. So if you want to create the ions, so these ions, when they are electrically charged molecules, as you know, are accelerated in an electric field. So their this speed depends on the ion's charge and the weight. So it depends on your ions both charge and the weight. So now how much time it takes for the ions to pass a certain distance, so this can be determined. So but it was not being able to perform on these large molecules or the biomolecules such as protein, because you can understand that it depends on both charge and the weight. So the large ions, so it will have a large charged ions which cannot be helpful to, which you cannot detect, or which was not possible. But these three scientists, which included your uh, Tanaka and this in the Nobel Prize, John B. Fain and Koichi Tanaka and Kututrich, which helped to show that the laser pulses could blast protein molecules so that ions in gaseous forms are produced. So this, so basic on the basis of their uh, in invention of the soft ionization techniques, it helped into the low energy, low soft ionization techniques of mass spectrometry to be used for uh, biomolecules like the proteins. Otherwise, it was not possible to identify and those determine those things. So this is one is your, so high hard ionization techniques is high energy, resulting in greater number of fragment and number of so molecular ion is low, but the for which the different proteins and other biomolecules can be used is the soft ionization techniques, which is a low energy thing, which have been developed by these three scientists and helped us to understand. So as I said that how so mass spectrometry, how it works is that it measures the mass by charge, and it doesn't measure your masses, but it measures your mass per charge. So what is this? So for example, if you have mass plus one charge, so after the ions you have ionized your mass, your sample or your peptide or anything, and if it is into generated into a singly charged species, so it will give you mass. So what will be your mass m by z that your mass spectrometer can detect is mass plus 1 divided by 1. But if you have a doubly charged species, like if you two charges are generated, then it will give the mass plus 2 by 2. So the more the charge or the doubly charged or the triply charged species you can generate, that will help you in identifying and detecting uh, the ions in that particular range. The M by Z, a small narrow range of M by Z. In the narrow range of M by Z, you can detect as many as you need to have the particular so you need to have a particular mass and those particular charges. So if more the number of charge species, the more narrow range of M by Z. So fully clear. So if it is a singly charged species, then it will have a very high ion species, and which we will require a greater range of mass analyzer to differentiate between those to detect that particular high M by Z, and it cannot be done easily in that particular narrow range of M by Z's mass analyzers. So these are the some of those particular mass analysis, like your one is the time of flight, which is that particular, as I said, that if you look at the block diagram of the if there's any question, it seems. What are the analyzers? So orbital and quadruple are itself analyzers. How do we separate the proteins based on? So we use liquid chromatography with changing the gradient of the hydrophobic solvent, and as we change the hydrophobicity, so similarly the proteins gets eluted. Yeah, so fully, and no more questions. So yeah. Yeah, so this is how your, as I said, that if you look at that, firstly your ionization area, then you will get into accelerated, the ions are accelerated, and you need to separate those ions so that your detector can detect those particular, and on the basis of the time it is taking to reach to the detector, and you can then calculate the Mascot charges. So this, how it does is that it separates your ions and measures their M by Z values based on the time they take to pass, or you can say that fly from ion source to detector. So on the basis of that, so either you will create the singly charged ion species or the doubly charged ion species, and then you can take the time to fly from the ion source to the your particular detector, and then you can identify the M by Z's. So this is your one M by Z, that is your uh, that is your time of flight. Second is the quadruple. So this is another kind of mass analyzer. What happens is that there are, you know, that there are two with the negative potential. So it, what happens is that it helps us a filter which allows us to only one certain of ions of a certain mass to charge ratio to pass to a filter to pass, and on the basis of that only can go. So people generally, so we cannot actually try to, so we will I'll go to that concept of the tandem mass spectra, otherwise we cannot detect the or derive the peptide sequence. So then these different mass analyzers are how are coupled, and then we can try to identify that what is the spectra, what is the sequence of a particular peptide. So this is another kind of mass analysis, which is the quadruple. and this is another kind of mass analyzer which helps to go through in separates the ions in an oscillating electric field. So here the ions are trapped in a kind of, and then it are released on the basis of the particular fragments and the precursors. So this is and this is in that it is another kind of where ion trapped, where the ions gets trapped. So all the things that we are generally trying to do is that to after the ions, a lot of ions are detected, we are trying to separate those ions. So first separation of after the peptides, when we get those ions, so now all those ions we need to let the detector to get identified. That what is those particular ions or the M by Z charges. So for this, we are taking in the second block after the ions are created. In the second block of the mass spectrometer, we are trying to separate those ions on the basis of the mass analysis, and finally, we are trying to detect those. Yeah. So as I told that what is the concept here is that if you are using, just try to understand that now that if you are using only one single analyzer, so for example, if we take TOF, you are using only TOF, and if you are using an ionization source as a MALDI or ESI anything, so if you take that, okay, you are taking MALDI as your ionization source. So you are giving your sample in a solid matrix, and then with the laser you are creating the singly charged species. So now your ions are generated, which is your ionization. Uh, then what you are trying to do is that you are accelerating it. So now is that once you have accelerated and you are trying to detect the, you are detecting, so you will get the entire M by Z of that particular ion or of that particular molecule. So you will can derive the molecular weight of that particular peptide or particular whatever, if you are giving any chemical compound or something, so you can try to go and find out. So after this, you will can derive the molecular weight of that particular compound or particular peptide. But you cannot. So what is the total molecular weight? But you cannot go and try to find out that what is the sequence of that particular. What is the sequence of that particular peptide? Why is it so? For example, so if we, for example, if you go and try to uh, if you try to go and try to find out that for example, if your sequence is for a peptide sequence is AP GHYK, for example, but don't, you can derive the molecular mass for that particular peptide sequence if you use only one mass analyzer, but cannot try to find out those peptide sequence, what is A or P or what is that, because you haven't fragmented it yet. As we mentioned that your next fragmentation, first fragmentation will give us that particular big charged ion with all the residues, and your second fragmentation, what will happen is that when you ionize, and then second fragmentation, what will it will do is that your sequences like the Y ions and the B ions, it will be generated on that particular amino acid or the peptide sequence, and then by calculating the Y2 minus Y1 and the B6 minus B7 and the Y ions and the B ions, then you can derive the mass of that particular amino acids, the monoisotopic mass. So generally from the basis of the mono, not only on the monoisotopic mass, but most abundant mass or the average weighted mass, because your amino acid or the elements like the carbon, hydrogen, oxygen, you know that they remain in many isotopes. So what happens is that you generally don't get mass of a single isotope. It's actually in the natural sources, it's the average of all the isotopes that remain present. So you can, with the difference between your y ions and the b ions, you can try to find out that particular mass of that particular amino acid sequence, and then by after calculating it you can derive that sequence of that particular ion. So you cannot for deducing the peptide sequence, you need to have a MSMS fragment. So without a second mass analyzer, you cannot derive the peptide sequence, but you can definitely understand the molecular weight of that molecule, or you can have of a compound or a molecular weight of a peptide, or that is also known as your peptide mass fingerprint. You can do that. So whatever the source ionization source be there, so the different mass analyzers, as we know, as we have discussed, like your TOF, time of flight quadruple. So generally you can TOF can be coupled with another TOF that will give you a TOF configuration mass spectrometry, or you can after quadruple you can have orbit trap, which is give you quadruple orbit trap kind of configuration that will give you after the fragmentation you will have a second fragmentation which will go to your detector, and with the MSMS spectra you can calculate those masses of those particular amino acids, those particular Y ions and the B ions, and can then deduce the peptide sequence. So you cannot have a generate a peptide sequence only using multi-top or multi-top. So that's the concept of the MSMS spectra or the tandem mass spectra. Yeah, so these two mass analyzers are required to have those hybrid configuration to identify the peptide. So here, so as I mentioned that when you have this, for example, this is a peptide sequence. Now you can, I think it will be clear for you that why we are choosing suddenly Y ions and the B ions and not any other ions. Like if you can see that it first fragmentation is the A1 ions and the X3 ions. So this is our R group of the of one amino acid. Then is our Y ions and B ions just after the C terminus and the N terminus, and then is the Z ions and the C ions. So what is detected? We are detecting are the Y ions and those particular B ions. So this B2 minus B1 will be that amino acid, and similarly this Y3 minus Y2 will be that another amino acids. So these at B and Y ions. So in ideally this, so as I was mentioning, I hope now it is much clearer than that time. So as I was mentioning that these two amino acids and these two are of the same amino acids. So you can see here, so this is the same amino acids. So these two ions getting fragmented, these two calculating those two masses will give us amino acid it belongs, and then we can calculating all those Y ions and B ions, we can derive the sequence of that particular peptide, which is generally done by the different software that are being used for the identification of the proteins and the peptides of the mass spectra, which we will look into in the sub. And try to identify what peptide and what protein samples we have used. So that's why this tandem mass spectra or the two mass analyzers are required to identify the particular peptides. Okay, so this is the kind of approach, as I was mentioning, is the bottom-up approach. Why bottom-up approach is that we have taken the particular protein. First, we digest using different proteolytic enzymes like trypsin or proteolytic enzymes, and then we are trying to get the peptide sequences, and then we trace back that those particular peptide sequences belong to which particular proteins, and then finally what we can do is that so what we can do is that what particular peptide sequences are belongs to which particular protein sequences, particular which proteins, and then can try to uh, identify that it belongs to which particular protein, and then we try to map the differential expression that okay, the proteins are belonging to that particular region, particular its their expression levels. We try to quantify first. We identify and then try to uh, quantify those levels at those peptides. So this is the bottom-up approach, whereas the top-down approach would be different in a way that the whole protein in that trying to find out. But here, what we generally use is the bottom-up approach. That after the peptide, if the ions are created, and then the fragmented MS2 fragments, we give the try to derive the sequence of those particular peptides. So this is the basic difference between top-down and the bottom-up approach.
Can you please repeat the top-down approach?
Top-down here, actually the entire protein is taken and not the peptides, and then that is only fragmented, and then those fragmented ions are coming from which protein, and all those things are trying to map. But it is not so much in use in daily cases. Here entire protein is taken, and here in the bottom up, so we are just, if you go by the name, so we are just breaking down the this peptide proteins into peptides, and after identifying those peptides, we are trying to uh, match it to the protein, further the protein sequence. So that's why bottom to up, and top down is that first entire protein and then breaking it to the subsequent fragment ions and trying to find out. So this is the basic difference between the bottom up and the top down. Okay, clear, right?
Yes.
Yeah. So how? So this is just an workflow. That entire workflow of how your bottom up MS based proteomics works is that firstly you will collect the different tissues and different populations. So whatever you can take, you will try to extract out the proteins. So you can have, you can so now those extraction of the proteins you can do also. You can take gel based or the gel free method. So gel based, if you separate out those proteins, and if you are looking at a particular protein, for example, you have identified, so take this example that you have identified one certain organism or certain uh, certain, you have identified certain new organisms, for example a bacteria you have identified, and it is you have find out that it is a novel bacteria. So what you have done, you have extracted the protein and have compared with another bacteria. So you have find out that all the proteins bands are exactly matching, but one particular band is not matching with that known bacteria. So you are interested in identifying that particular protein for that particular band what is coming. So you are just taking out that particular band from the gel, and then you are doing the in gel based digestion, and then trying to find out that identify that what the sequences of that particular peptide or for that particular protein. And another is that in solution, after you done the uh, in you can, another method is that reverse phase in the HPLC columns using liquid chromatography. You are fractionating your particular peptides on the basis of certain pH. And other thing is that the shotgun proteomics, separation of the extracted protein, where you are not doing any gel based or gel free and directly injecting it into the uh, uh, RPLC column, and then you are trying to separate it there only using the gradient. And once you are done, then you are trying to inject it and create the ions. So after you do the protein digestion, so you will use in the reverse phase liquid chromatography with eluting all the peptides, hydrophobic peptides, and then you will try to find out the mass spectrometry, both tandem mass spectrometry analysis, where you will get the peptides M by Z values, calculate the sequences, and then try to map it with the particular proteins. And for that, after finally, you will try to use different tools and bioinformatics based things. You will try to identify those proteins, and then you will do further analysis, statistical based analysis, to identify which proteins are differentially regulated, and then the pathway analysis and all. So this is generally how a bottom-up MS-based proteomics workflow generally uh, takes place. So here digestion is being done with through the enzymes to generate your fragments. So because you need to have fragments within the range of M by Z, so that you can try to find out, detect in the ions. Otherwise, it won't be possible for you to detect. And for example, so what proteolytic enzymes, so mostly commonly what is used is your trypsin, which cleaves after lysine or arginine, except if proline is there and it does not let the cleavage to happen. Okay, so this is a bottom-up MS-based proteomics general workflow. So now, as we had, so I think now it is much more clearer to understand how it is happening. So now, if we go for the quantitative proteomics, so for example, the level-based proteomics. So as I told, so as we have discussed, ITRAC, the isobaric tag, the relative absolute quantifications. Another approach is the TMT tag, tandem mass tag. So here, ITRAC is that you can use only 8 levels, but here you can go to 16-plex. So what here is that the multiplexing is the key in this kind of level-based approaches, where you can use a number of samples and try to identify their groups in a single runs. And this helps in another way is that if you have a protein sequence and if you fractionate it, those different sequence fractionated to increase your coverage, then you can use those TMT based approaches that all those conditions are 16 conditions are fractionated, so it will take so much run. But if you pull it, so each fraction can be run at one go, which will help you to get those in a very much multiplexed way to identify all those fractions conditions in a single run. So this is how the TMT also works in the same way, where your NHS this sequence gets attached to your particular peptide. So after you digest your peptide, you will label those, and then the reporter ions you will try to identify in the MS quantitation. So this is the concept of your TMT. So another in the label-based proteomics, in the cell culture-based experiments, if you are working in the cell culture-based experiments and want to have proteomics from their particular cells, so one is your CLAP or stable isotope labeled affinity, and one is your ICAT. So what here is that you in the cell, when you are growing your cells or maintaining your cells, so what you will do is that one cell condition. So for example, you are trying to find out between a cell having a particular treating, you are treating with a particular drug, and another with not treating with a particular drug. So cell culture we will one we will do with the normal arginine, another one we will do with a heavy arginine. Then you will combine those cell lysates and then purify proteins, and then you will try to find out in the say tandem MS, and so the light peptide and the heavy peptide, and these are you can quantify. So all this in that ICAT also you will uh, light to light linked and the heavy linked H1 and the H2, and on the basis of that you can quantify. So all these are help us to understand the absolute in the kind of a relative quantification of those particular proteins in a particular state, either in between a cell or in a cell state or in a disease state or in a healthy state. But all these are not your kind of an absolute quantification. These are all your examples of the relative quantification. So if you want to go for an absolute quantification, you have to have that peptides and try to find out in a preparation of a standard curve and those using the different synthetic peptides, and then only you can go for an absolute quantification. So this is for the level-based approaches, some of the level-based approaches that are being used for understanding and identification of the peptides. And in the level-free quantification, as I said, that if we do the digestion of the peptides and then we try to find out the LCM SMS, so these are the peak intensities we get on the basis of the spectral counts, that how much the strong the current is. Peptides is much more in number in the samples if you try to visualize the thing. So the ions generated for that particular species would be more with those M by Z, and the current that will generate it will be much strong in the detector. So the spectral and the spectral count will also be much. On the basis of that, you can understand that in this particular sample, this particular peptide has your much more intensity value than the other. So in this way, you can find out using the level-free quantification without any level. So here in each single run, you will try to find out those particular proteins, peptides, and then can identify those. So this so another concept in the MS based proteomics is one is your, after all this is your DTA and DIA, that is your data dependent acquisition and the data independent acquisition. So what is the we are so if we just now recall the concept, what we are doing is that creation of the ions, and then what we are doing is that uh, generation of those fragment ions. So what we are saying is that if we try to identify using the peptide sequence, so what we told that we need to have a in a work in a tandem mass spectra, where MSMS spectra we need to generate to reduce the uh, sequence of the peptide. So now what we are doing is that once the fragmentation is done, then what we are doing is that we are scanning those particular sequences where the top for in the DDA method or the data dependent acquisition method, we are saying that the for this particular fragments top 10, top 20 or top n, to say top n abundant fragments or intensity only you take forward for detecting in the after for detecting in the detector. So this is the data dependent acquisition. We are saying that take down the fragmentation of the selected precursors, that the top 10 or the top 20 you take, and then only you try to identify in the MS2 level. So MS1 will get the survey scan and the precursor selection. So once the fragmentation is done at the MS1 level, so what we are doing is that we are selecting the top n fragments of those MS1 level, and then we are sending it in the MS2 level to get detected. So this is what is known as the data dependent acquisition, where we are saying the instrument to select out those particular top fragments, and not the all fragments to all the fragments to get monitored in the detected. Whereas in the data independent acquisition, what is happening is that we give a scan range or a range of M by Z, for example 200 to 800 M by Z or certain M by Z, and all the fragments that come within that range is getting detected in that particular range, in that detector, in that particular range. Whatever it gets filled, it will get detected, and it will not look at that which one is abundant, which one is not abundant, and top 20, top 20, no, just on the basis of the range. So this is another in the tandem mass spectra. This is another concept. One is of data dependent acquisition, another is of the data independent acquisition, which leads you to the identification of those particular proteins. Am I clear, right? No question. Okay.
Sir,
Yeah.
So in which case are we going to use what? DDA we are going to use when, and DIA we are going to use when? How do
So
we know
this
that for
no,
this
so
case
this,
we'll use that?
so this is depends on your experimental principle, experimental requirement. So if you know that organisms library which you know, and you know that your fragments, the proteins you are looking at for, and the fragments comes in this range of M by Z, then you will not you will try to detect all those fragments. Then you can do a DIA kind of thing. This is just one example. And if you have no idea and don't want to that in which range my peptides and the proteins can go, so then it's better to go for the DDA. Yeah.
Understood, sir. Thank you.
Yeah. Any other question or anything?
So can't we use gas chromatography? Is it necessary to use LC?
Gas chromatography has other what you say that in the metabolomics and matlab in the small compounds and all those generally it's used, but for the peptides generally LC MSMS based techniques are used.
Okay.
Yeah, so these are other concepts of the resolution, isotopic peaks, and the charge state. So as I was mentioning that our peaks, our elements, the carbon, nitrogen, and the or that doesn't remain in a single isotope space, isotopic thing, but generally if there is abundant and in the average weighted thing. So the M by Z difference that between that helps you to identify the M by Z as small as level, that is called your resolution, that you can identify between the two isotropic peaks, or the two different peaks, two different peaks of the same masses, of the different masses, the small whatever small mass differences is there. And if your mass spectrometer is able to identify those particular two differences, then it is a good resolution. So how we can uh, see that? So let us look at that. M by Z difference between isotopic peaks is 0.25, and mass difference is 1 mu with 1 neutron, and then you can see that Z is the fourth charge. So monoisotopic will be generally it is calculated accordingly. So now if you see that for example orbitrap mass analyzer having a resolution of 60,000, you can see that it is help to resolve your 431.73, which is your monoisotopic peak, 431.98, 432.23, 432.28, 430.73. It can detect the small differences as well. But here generally it is when you have a low resolution, then you can detect mostly the one in the 432, and you cannot get all the four peaks in a you cannot detect those all those monoisotopic peaks in a very good way or good manner on the basis of the resolution of the instrument. So what is that resolution? And it is a ratio of the m divided by your delta m, which is at the half maximum of the intensity, and at its full and it's at the half maximum, what is its width. So what it how it is calculated is that it is calculated at the half maxima, what is its maximum what is its width. So this is the mass. So for example here if it is around 15 point something, so this divided by the width of this, so this gives us the, and this is also the delta mass between the 2 points. So the more narrow it is, the more the peak is narrow, the better is the resolution of that particular instrument, and the better is that we can try to identify, we can try to resolve the mass differences, such small mass differences between those particular peptides. So, sorry, not particular peptides, but particular ions and the mass charges. So, the more the better the resolution, the more it will be narrower, and more we can we can try to identify those. So, this is how the mass resolution is generally calculated. That at the half maxima, what is its width? So, for example, for this mass intensity, we are getting 100. So, what is half maxima intensity is 50. So, what is the width at this region? So, 100 divide sorry 100 is 15 divided by your width would be giving the mass by delta mass, which is your particular resolution. Okay. So another is the mass experimental mass. So what happens is that it generally what is the we don't get for the theoretical mass, but what we can get is the uh, observed mass. So what is the difference between the observed mass minus the theoretical mass? If the exact theoretical mass is 150 and we are getting something 149, so what is the observed mass minus the theoretical mass divided by theoretical mass, and it is generally calculated in the ppm. So the for the instrument, so if it is less than 1 ppm, then the error rate for that particular instrument, for that particular mass spectrometer, to calculate the masses accurately is so good. So it should be ideal case, it should be less than 1 or less than 0.5 ppm. But different instruments come with your different, so I'm not going into the comparison for the different instruments, but it depends on the accuracy, depends on the your the different mass spectrometer. So what is the so if you get the after different experiments, so what is your observed mass and minus theoretical mass, it will give you the mass accuracy. So these are the two things. So how well your instrument is able to resolve your two monoisotopic peaks or two peaks with the very different masses and the embasses, and what is the mass accuracy, and your resolution is always calculated is a on the basis of that in the half maxima, what is your width of that particular mass. On the more narrow it is, so more it will be higher resolution your instrument would be. So another just to give so in the whole structure, if you can think of that, we can understood that how the ions are created and then how the mass analyzers are helping in the separation of ions. So now how, yeah. Hello, I'm audible, right?
Yes, sir.
Yeah, audible. Okay, fine. Yeah, so these are the two different dissociations. So how these ions are generated in inert gases are generally used. So each one is your collision induced dissociation. Audible.
Yes, you're audible.
Yeah. Yeah, so this collision induced dissociation, what happens is that using the gas phase gaseous molecules, these precursor ions, these product ions are generally created, and which further leads to the MS2 level. So these are mostly two types of dissociation: or collision induced dissociation. Another dissociation is electron transport dissociation, and another there is the HCD, which is called before the seed trap in certain instrument. But mostly the principle remains of the CID, or the collision induced dissociation. So this is another is the electron transport dissociation. As I told that with electrons, these ions are generated, and then which is being taken forward for to identify the uh, ions for further analyzing in the spectrum. So these, so mostly the CID cells are generally being used for generating those particular product ions, and the peptides on the basis of their class charges, it's are generally taken forward and uh, analyzed in the spectra. So this was for all those different ionization techniques: mass resolution, mass accuracy, and then is the uh, what you told is that the different mass analyzers and how it is done. So any confusion or any doubt till now? I hope you are clear, right? Why tandem mass spectra, and why it is used, and what is that MS1 and the MS2 levels. Okay. So now if we go to try to look at some of the calculations of some of the things. So these are simple basic things. So I just wanted to give a flavor that how these different calculations are being done. So for example, if we take the TOF based mass spectrometer, where the analyzer is the time of flight. So what we know is that this is a simple concept. The time, so velocity equal to distance by time, and from where we can derive the time. So in meter per second, then we already know that the if we go for the formula of the kinetic energy equal to half mv square, and from here we can get the velocity as well, root 2 ke by m. Now if we just substitute that t, so it will be d by root 2 ke by m. So we will get t equal to d m by 2 ke. Okay, so this is the formula for the calculation of the time, and d by m by 2 ke. So now if the question comes like this: 1 mole of 24 mg plus ions, so with for those mass has a mass of 24 gram per mole. So we need to divide. So how we will divide it? So we will divide by the Avogadro's number to get the total mass of that particular ions, 1 mole of mass. So now if we try to calculate that what is the time required for those ions to get to that detector, so you can just and we need to know the length of that as well. So the questions can be either you can have to derive the name, the derive the length if you want to reach in a particular time, or vice versa. So and then you will write the masses and the kinetic energy, and then you can easily calculate the time required for your ions to fly in the TOF. Okay, so the simple equation of the kinetic energy. So since the ions are accelerated in your electric field, so with your kinetic energy, you can calculate the time required for to fly. So then is if you want to calculate the masses and the kinetic energy. So generally the mass is your relative isotopic mass into 10 to the power minus 3 divided by Avogadro's number for the particular Avogadro's number will give you the mass. So if you a question comes like this, that SR plus ion travels through a top of a mass spectrometer with an energy of this much. So this is your kinetic energy, and then the ion takes 1.09 into 10 to the power minus 5 seconds to reach the detector. Calculate the length of the flight tube in meters. So this you can try out on your own that whether you are getting the answer. So I have mentioned the answer. So what step 1 you will do is that you will calculate the velocity of the formula root 2 ke by m. So kinetic energy is by given mass. You can understand that with 86 into 10 to the power minus 3 by 6.022 into 10 to the power 23 will give your mass. So you can calculate the velocity, and then you can get the distance and the time. Using that, you can calculate what is your length of the flight tube, what is the length of the flight tube for this particular ion. Okay, so these are simple some sums which you can try to identify. So now we will go on to find out some of the hands-on, like once you have, this is uh, one of the software where we can try to identify the peptides. For example, you have taken a sample and have run in a mass spectrometer, and you in the mass spectrometer you have got a file. Now if you want to find out that what those sample belongs to, so this is one simple example. So here with so it generally takes an mgf file, that is your mascot generic format file. So with that you can easily go and try to do that. So also along with me also, you can all can go and try to see the parameters and all, and how it is done. So if you go and, so this is a kind of an hands-on software. You got the theoretical aspects and the theoretical understanding. So now how we can identify that particular peptide or a particular protein in an actually samples. So after we go here, so we will do is that Mascot database search. Okay, so now here what is there, you can find out there are two things. One is the peptide mass fingerprint. So as I told that if you run your sample in a way that it is your coming in a sample in a MS based, and do not go for the MSMS based spectra or the MSMS based search, then you will, what you will find out that the molecular weight of that particular peptide or particular weight of that particular compound, but we cannot get the derive the sequence. So that's why we are not going for the peptide mass fingerprint. Rather, we will go for the MSMS ion search. So this MSMS ion search will help us to understand the calculate the peptide sequences, derive the peptide sequences, and then map with the particular proteins, and from the particular databases that are available, like publicly available databases like UniProt or the SwissProt, and then we can get our data. So if you go and search here, okay, so this will come up. So if you just put my name, you can put your name. So this is your you need to have a given genuine email ID, because if it is not being, you have not activated it, it will require you to activate it. So what you will do is that search title you can give anything. So I'll just give Proteogenomics week 3. Yeah, so the databases. Now here are the options. So just look at these parameters, that how you can do. So for example, if you want to add these databases, so what is this contaminants? So when we prepare the samples, so there are different contaminants like some abundant proteins like keratin and all, which comes into naturally from the environment into the samples, and which is not actually from our sample but rather the contaminants. So this contaminant database helps us to identify those peptide sequences or those particular protein sequences which are not from our originally samples and can be removed from the samples. So this is one. Second is that if you, we need to identify those particular peptides are coming from which organism. So if you know that it is from your COVID you are doing, or you are doing from C. elegans or yeast, Saccharomyces, or any kind of organism, or Homo sapiens, or Mus musculus, or rodents, or anything. So then you can use that particular sequence or the particular database which will lead you to use that particular sequence only. But if it is not, then what you can do is that we will use Swiss plot. Swiss plot contains the protein sequences of all the organisms. So you can naturally use that. So whatever organisms it is, it will get detected. So we don't need to have any particular difficulty in we do not need to, since we don't know that our file contents of which we have done using a blinded means not knowingly which organism, so we are taking Swiss plot. So taxonomy will be all entries. Enzyme we are using trypsin. So if you just go to the drop down, you can see all those proteolytic enzymes that are used, like trypsin, uh, trypsin P. You can use that as well, that without proline, and then chymotrypsin, lyse, and all those things. So we will use trypsin. Allow up to miscleavages. So what is the concept of your miscleavage is that you think that the trypsin is not efficient enough to cut at all your lysine and arginine, and there might be some where it has left over, and so here we generally what we use here, allow up to the 2 missed cleavages. Then your quantitation. So what we have just discussed, the quantitation, you can get all those iTrack and all CLAC. So let us none means level free. Cross-linking also comes with that. If you have done any kind of cross-linking or not, it's none. Then comes the fixed modification. So fixed modification is that one which generally happens during our proteomics digestion or the sample preparation step. What we do is that we generally do is that there is a reduction step when we are doing a digestion from the proteins to the peptides. So why there is a reduction step? Because the proteins, you know, that remains in a folded state, or the so the trypsin cannot affect or cannot help you, cannot go and cut at every position. So you need to open up that particular protein and need to take it at as a particular peptide. So what happens is that when the peptide is open, so on that basis, when during the reduction there is a extra modification gets added, which is your carbamidomethylation at the cysteine residues. So you will add this as fixed modification, and then so this will generally give. Now for the variable modification. So if variable modification is that one, if you want to look for the PTMs like glycosylation, then you have phosphorylation, acetylation. You don't know that whether it has happened or not. It is not fixed. So you will give the it as in the uh, uh, variable modification, that it might happen or it might not happen. Because if you once you give at the fixed modification, then your algorithm will look at for the modifications, and if there is it doesn't find, it will not calculate it. So here we will give a, let us try with giving the oxidation at the methionine. Okay, so next is that you can give change the peptide tolerance, the number of 13C, as I told, isotopic and all. So let us keep at so you can the peptide charge, there's the singly charged, the doubly charged, all those. So let us keep at 2 plus, and then we'll choose a file. So as I told that it will be in a MGF format, a Mascot generic format. Okay. Also you can have an MZML, MZ data, and all this format it takes. So we are giving in the Mascot generic format. Instrument you can, as now I think you all are familiar and you all can easily understand. So you can see that ESI quad top. So as I had mentioned that you will need a tandem mass spectra works on combining the two mass analyzers. So here you are getting like quadruple top or multi top PhD. So on the basis of that multi top, so you can understand. So we are keeping it as a default. We are not keeping anything, and then we are doing is that starting the search. So now let us see that how which peptide sequences is get matched. Yeah, so now you can see that there are three, it has given three proteins. One is your chlorophyll AB binding protein of Arabidopsis thaliana. Another is your isopropylamide dehydratase, that is also of Arabidopsis thaliana. And another is the chlorophyll AB binding protein CP26. So all this, so these are given on the basis of the peptide. So for example, if you are getting a shared peptide in among all these three proteins, then it will give the name that it can be from that particular protein. But on the basis of the score and how many peptides have been matches have been done, or on the what is the coverage percentage, then you will get that how many what is the actual protein that you are getting. Okay, so this is, you can see that there have been for this particular of that score is 238 and their scores are 45 and 41. You can clearly understand that it is quite less, and these are the 6 peptides have been matches. So if we go to it, yeah, so before going to that, if I, yeah, so here what I would like to mention here is that here you can see that it is the chlorophyll AB binding protein 2. The most of the peptides have been matched with this. This is the score. This is your monoisotopic mass, and this is your carbohydrate. This is your enzyme trypsin cuts C-term side of KR unless next residue. So what is your sequence coverage? That is your 52%. Okay, so these are the peptide sequences and every details you can get. So I have told you about the coverage. So whatever more coverage you will get, so you will know that okay, the more likely is that your peptide sequences to be from that particular protein. So this is important to get identified. Next is that what you have done is that so if I go here, so let us take a big sequence so that more variance and yeah. So as I told, as you can see in the theory that we have this peptide sequence and this Y1 and the B. So you can see that A, B, B2, B4 and all those sequences. So what are those sequences? What ions have been identified to make these particular sequences? You can visualize here. So as I was mentioning in the theory, you can visualize it here that how it has been identified. Now if you go here, so these you can see that these are the y ions. So these y ions minus these y ions uh, uh, and calculate trace back. So let us look at another one. Yeah, so for example, if you go here, so here y ions minus this minus this should be your this, and also you can see that it has been b ions have been detected. So if your y ions are detected along with that, if your so as we mentioned that generally the b ions are less abundant because of the proteolytic enzymes that we use. But if the b ions are also generated and the y ions what we detect y1 uh, uh, y1 minus 1 2 and the b n minus 1 minus n, if it is same, then it is also a type of another validation or another type of thing that okay, so this particular amino acid is correct. So in this table you can identify that this red shows that what y ions fragments have been identified, and by doing a minus you can get the monoisotopic mass and then can derive that what is the sequence, peptide sequence. And once you derive the peptide sequence, you this you will, and you will map with the particular protein. So you can see that with the SwissProt database it could map with the chlorophyll AB binding protein. So your sample contained the protein of the chlorophyll binding protein AB binding protein was your sample. Okay, so these are the different parameters, and this is how you will interpret the spectra. I hope it has been clear to all of